Good morning, and thank you. Thanks for the gracious introduction, Kathy. I listed a bunch of affiliations here this morning because I'm wearing my NIST hat. NIST is the U.S. Department of Commerce's agency that does measurement science and standards. What we do is act as the nation's measurement institute. When I wake up in the morning, it's my job to put in infrastructure to make the world safe for bioeconomy, make the world safe for advances in biomedical instrumentation measurements that do make it into practice in our economy and in our society. So that's my first position. Those are the guys that write me a paycheck. Hopefully that will continue past Wednesday night at midnight. John Boehner has thrown himself on the fire to try and dampen that down this morning, and that's awesome. I have an awesome time here also leading a team that is trying to figure out how to work collaboratively with the academic efforts at Stanford University, which are, I've been out visiting Stanford University since I've been working in biology since the sort of early aughts, 2003, a world-leading site for creative, curiosity-driven research, but also such a powerhouse here, and getting stuff over the fence and picked up by commercial practice, picked up by the biomedical practitioners. So it's great that I have collegial appointments as a consulting professor in both the bioengineering department and the pathology department. And finally, I've been invited to be a participating faculty in CIRCE. And really, that's the hat I'm going to be wearing this morning throughout this conversation, is what does mission-relevant research look like in regulatory science in today's world of emerging, quote-unquote, next-gen diagnostics. David did a great tour of what FDA is trying to do in implementing policy that would be agile and adaptive and, I forget the other word, but accommodating. Accommodating, is that a good word? Anyway, this is me as an academic today. How do we, and my job, frankly, is loops back everything, the snake eating its own tail. My job is to ask, how can I make sure that the innovation makes it over the fence and into practice, that quote unquote, and I don't want to use this term loosely, but I've heard the term, so I'll use it, regulatory barriers to adoption of new technologies. So that doesn't really happen. Does NIST play a role in establishing the infrastructure that lets this stuff make it into practice? So I'm wondering, are there, riffing on what FDA have proposed around using standards as a basis for establishing safety and efficacy, what standards ought there to be? Who ought to make them? Liz Mansfield hosted a panel back in February of this year at an open public FDA meeting at NIH, and I was on that panel and I was asked, so who should make these standards? And so this is what I said, basically. You actually need a whole architecture of standards. There's not like one body that's going to make one set of standards that can be used to evaluate the safety and efficacy of a next generation diagnostic. Because this is what it looks like. And this is, David, this is my nonlinear form of the flow chart that David showed. And it is my snake eating its own tail, because really every aspect of this process, where you go from pre-analytical to sequencing to sequence bioinformatics to clinical to functional annotation, to clinical variant knowledge-based queries, to clinical interpretation, reporting, and archival, every aspect of this affects every other aspect. And so we don't have complete independence or orthogonality, but we have opportunity to make them all interoperate. Kathy gracefully introduced the work we've been doing. Justin Zook co-leading with me, my colleague from NIST, who's here with us this morning, co-leading the Genome in a Bottle Consortium. Many of your faces I recognize as welcome colleagues in that consortium, where what we're doing is, and this is my linear version of the process, what we're doing is trying to develop whole human genomes well characterized for their variants against the reference genome that can be used to give you what is essentially a stoplight diagnostic, not to overload the word diagnostic, a stoplight go, no go on your measurement process. So at your, the most valuable piece of this is if you can't get the right answers on the genomic DNA we supply that comes along with its variants, there's something broken and you get a red light. If you get the right answers, you get a yellow light to proceed with caution. There's no green light from running a dead control, essentially, DNA in a tube. And in fact, there are limitations more significant than just that you can't be certain about your clinical sample here, but we provide extracted whole human genomic DNA in a tube with probably a median or most likely fragment length on the order of 60 kilobases. It may be not as interesting in 3 years if things really go the way they're going and we get longer reads that are not covered by our reference controls. But we're trying to make available NIST reference materials to support this. And there's some details on this on the side, and I'm happy to talk to anybody more about it at the break, or as would Justin be. But going back to this overall enterprise process, those two boxes that are highlighted here is what those genome metabolic controls are good for. It's not going to help you understand or demonstrate the performance of the pre-analytical piece. It's not at this point, although it's the next frontier for us, along with getting at the more difficult regions of the genome and getting at the more different classes of variants, it's not going to tell you whether or not you're functionally annotating or establishing the molecular impact of the variant in any proper way. We'd like to do some gold standard, quote unquote, functional variant annotation on the genome in a bottle samples, but we haven't gotten that underway yet. Really interestingly, I think there's great regulatory science advance to happen in much of the rest of the process. How do you establish that a knowledge base is valid? How do you not only have processes to establish that the data are meaningful, but how can you assess the completeness? And are there some true scientific advances that we can make in understanding what's there and its veracity for the application you're subjecting it to? And similarly, clinical interpretation is an area where there has been little science done to underpin confidence in the clinical interpretation of a properly functionally annotated and well-characterized variant for therapeutic decision-making or clinical grade decision-making. So I think, so this is again me wearing my hat as a CERSI faculty, these are the kinds of unmet needs and knowledge gaps in such a, in the new frontier. So awesome opportunity. I did want to say that, in fact, we're grateful to be able to be issuing reference materials. This is, our development work has happened in no small part in both substantive and spiritual partnership with our sister agency at the FDA. A lot of the funding for the work that we've done in my lab to do the genome in a bottle samples, and a lot of the initiative to carry out building the first human genome reference materials happened in partnership with FDA support in, and again, both the spiritual and the material realms. And this is a nice, it's the first time I've issued a reference material, and I've been at NIST 25 years, I've been issuing a lot of reference materials in that whole time. First time the New York Times thought it was worthy of note. This is also the first time where the innovative work to develop the data around that reference material made it to really high-profile publication in a Nature Biotech paper, and this is Justin's paper that described our arbitration process whereby we got the gold standard variant calls. This has all enabled us to really invest in characterizing the next set of reference materials. So we have 6 new genomes, essentially 3 new reference materials coming out, 2 trios that were part of the Personal Genomes Project that have been characterized with a dazzling, I would say, dazzling suite of analytic methods, sequencing methods that are enumerated on this list. Most notable is that we have one of the first trio samples with open public data using the PacBio long-read sequencing to really characterize that. And you can see that we've got on the order of 70x coverage on the son of the trio and 30x on each of the parents. We're looking forward to the analysis, which is underway in at least 15 groups around the world of these unique data in order to come up with a more complete suite of genomic reference samples that, again, are really only going to be good to give you a yellow light if you get the right answers. But that's our strategy. Of note, the data are open and public. No embargo. As we measure it, as we have confidence that, in fact, the file names are right, we get them out on the FTP site. And a bioRxiv paper which has also been submitted at Nature Scientific Data, is in fact up there. And I'll note that it's also the first paper we've had that in preprint form has been tweeted more than 100 times. So we're excited about that. We just had a workshop in August. This preview piece came out in Cell Systems yesterday or Wednesday. That describes the workshop and puts in context the Genome in a Bottle project with some other public data recently released out of WashU that is for some cancer genomes. I'm going to whip through a table which is also going to be available as a slide deck here that I've built describing the characteristics of the standards at each level and describing, most importantly, the knowledge gaps. So these are the titles of the columns of the table I'm going to walk through with you. It's a table in PowerPoint. Good morning. Death by PowerPoint will proceed. But the columns are, what evidence do you need to develop at each of those layers in the process? What sort of standards or evidence developing practices are appropriate to develop that evidence? Who are the example stakeholders that might be engaged in the development of the standards or the best practices? And finally, and this is again me wearing my CERSI hat, what are the knowledge gaps? What research agenda ought to be undertaken to fill those knowledge gaps to make this real, so that not only can we do analytical validity against standards, but we can, in fact, carry forward and understand the safety and efficacy of next-gen sequencing as a clinical test in a comprehensive way. So this is what our table is going to look like. This piece is hard to do, and people have been taking whacks at it in best spirit, but when, this is about the pre-analytical. What evidence needs to be established in a pre-analytical to establish that the pre-analytical processing is valid? You need to demonstrate you're doing representative sampling of the genome under study. If that's a tumor sample, then you need to understand how representative that sampling is. You need to demonstrate that you've got accurate extraction of DNA or unbiased extraction of DNA. If it's an FFPE sample, you're going to have to demonstrate that you have a meaningful extraction at least, and that the DNA is meaningfully representative of the clinical application at that point. What's the integrity of that DNA? So the kinds of things I identify, and again, this table is by no means a recipe. This is, again, me wearing my more academic hat. These are the kinds of ideas that need to be sat down and worked at. Probably we need a workshop to spend 3/4 of a day with the right people in the room, and clinical labs, clinical standards development organizations, professional societies, looking at the questions of, wait a minute, what goes into a documentary standard that talks about pre-analytical? What sort of reference samples would support confidence in the pre-analytical process? And finally, there's an example knowledge gap at the end that I already mentioned. Similarly, you do one of these tables for sequencing to go from the DNA to raw sequence data. And you've got to understand how or whether it's unbiased. You have to understand whether it's appropriate, fit for purpose. If you're doing 30 base reads, you've got to demonstrate that those 30 base reads can in fact be used in a manner that can be diagnostic for the condition you're claiming diagnostic capability, and so on. I'm not going to read the tables to you. Again, these are available, but there's knowledge gaps in each that I'm trying to establish as a research agenda that I think CIRSE might be a great organizing principle to carry out that research agenda. I'm not sure that there are CIRSE investigators in this CIRSE that are going to be the right scientific leads, or whether it's a bigger network, but I do think that it can be the convening body. It's my hope as a NIST person, as a Stanford person, as a CIRSE person, it's my hope that somebody takes this on and that kind of research agenda is established in an open, transparent way, and it's the best of the best. So we do the same thing for raw sequence data to VCF, or what I call sequence bioinformatics, and you can sort of see the knowledge gaps. You can see a quick pass of the example stakeholders and the kinds of evidence that needs to be developed. And you can do the same thing for functional variant annotation, which is this VCF to a molecular impact. Is that a truncating variant? Is it a synonymous variant, et cetera? Is this a regulatory variant? What's its likely impact? Can it be carried forward into a clinical knowledge base? And there are some awesome research questions here. How do you, that completeness question is a really good one. And standards do offer, I think, and this is my Department of Commerce hat, I should have had 3 hats up here, standards really do offer us a way so that such a database could be kept private, because you can demonstrate through consistency with standards or with standard manners of evaluating that your private database meets certain criteria. Clinical interpretation, again, this is where it gets even a little fuzzier of how do you develop a standard of clinical interpretation such that you can demonstrate the quality and the reliability, the safety and efficacy of the result. Reporting may be more simple, it may not be, and this would, again, lean on the professional societies, I think, to establish practices, and again, offers opportunity for thoughtful development of standards. And EHR archival is, people should care about this, and I think they do. It's another place where the CIRSE can convene and make that happen. So I'm gonna finish up with just putting that architecture of the process up here, and offering up that there is underlying this whole vision for standards-based regulatory oversight, great opportunity to really feed the whole next generation diagnostic, not only DNA sequencing, but everything that one can imagine that fits into the description David gave earlier on these next-gen tests where you don't have a single analyte, where you don't have a single indication, where you don't have a single outcome. I'm thrilled to be here. I'm really grateful to see the engaged audience, and thanks for the opportunity. Happy to take questions.